Right, good evening and welcome back to my routine lectures. I'm Dr. Bobby and this is from New Age Cosmetic Wired EdTech Healthcare Consultancies. Welcome back. Continuing on yesterday's topic of cell organization, we are on the section D3, cell cycle. Fundamental concepts of cell cycle. The cell cycle is the sequence of events between successive cell divisions. Many different processes must be coordinated during the cell cycle, some of which occur continuously, example cell growth, and some discontinuously as events of landmarks, example cell division. Cell division must be coordinated with growth and DNA replication so that cell size and DNA content remain constant. The cell cycle comprises a nuclear or chromosomal cycle, DNA replication and partition, and a cytoplasmic or cell division cycle, doubling and division of cytoplasmic components, which in eukaryotes includes the organelles. The DNA is considered separately from other cell contents because it is usually present in only one or two copies per vegetative cell, and its replication and segregation must therefore be precisely controlled. Most of the remainder of the cell contents are synthesized continuously and in sufficient quantity to be distributed equally into the daughter cells when the parental cell is big enough to divide. An exception is the centrosome, an organelle that is pivotal in the process of chromosome segregation itself, which is duplicated prior to mitosis and segregated into the daughter cells with the chromosomes, the centrosome cycle. In eukaryotes, the two major events of the chromosomal cycle, replication and meiosis, are controlled so that they can never occur simultaneously. Conversely, in bacteria, the analogous processes, replication and partition, are coordinated so that partially replicated chromosomes can segregate during rapid growth. The eukaryotic cell cycle is divided into discrete phases which proceed in a particular order, whereas the stages of the bacterial cell cycle may overlap. The progress of the eukaryotic cell cycle is controlled at checkpoints where regulatory proteins receive input from monitors of the cell cycle itself, intrinsic information, and monitors of the environment, extrinsic information. Intrinsic monitoring ensures that the stages of the cell cycle proceed in the correct order and that one stage is completed before the next begins. Extrinsic monitoring coordinates all cell division with cell growth and arrests the cell cycle if the environment is unsuitable. The cell cycle is controlled by protein kinases. Cell cycle transitions involve positive feedback loops which cause sudden bursts of kinase activity, allowing switches in the states of phosphorylation of batteries of effector proteins. Cell cycle checkpoints are regulatory systems which inhibit those kinases if the internal or external environment is unsuitable. The alternation of DNA replication and mitosis is controlled by negative feedback. Mitosis is inhibited by unfinished DNA replication, and DNA replication is prevented during mitosis by the phosphorylation and inactivation of a protein required for replication. The cell cycle is a result of a complex network of information in which kinases are controlled by the integration of multiple positive and negative signals. The bacterial cell cycle. The DNA replication and growth coordination. The Helms-Tedder-Cooper model, or 1 plus C plus D model, divides the bacterial chromosome cycle into three phases. The interval phase, the chromosome replication phase, and the division phase represented by the letters 1, C, and D respectively. DNA replication occurs during the C phase. Its duration is fixed, about 40 minutes in E. coli, reflecting the time taken to replicate the whole chromosome. The D phase begins when replication is complete and culminates in cell division. The duration of the D phase is also fixed, about 20 minutes in E. coli, and can be regarded as the time required to synthesize the cellular components required for cell division. The minimum duration of the chromosome cycle in E. coli is thus 1 hour. Because C plus D is fixed, any change in the cell doubling time must reflect a change in the duration of 1 I should read it as I. The interval between successive initiations of replication. The doubling time of E. coli can be as long as 3 hours or as short as 20 minutes. During slow growth, 1 is greater than C plus D and the replication is completed before cell division. During rapid growth, however, the doubling time is shorter than the time taken to complete a round of replication and cell division. The only way the cell can accommodate its fixed chromosome cycle into the accelerated cell division cycle is to make 1 less than C plus D, that is, new rounds of replication must begin before the previous round is complete. Therefore, during rapid growth, daughter cells inherit chromosomes which are already partially replicated. Multiforked ozomes so that replication can be completed before the next round of cell division. The frequency of initiation is thought to be controlled by a positive regulator which must be present at a certain critical concentration per origin of replication QV for initiation to be successful. During rapid growth, the regulator accumulates more quickly, allowing more frequent initiation. Once initiation has occurred, the number of origins in the cell doubles, and the effective concentration of the regulator is halved, so that it must accumulate again for another round of initiation to occur. The existence of a positive regulator is predicted because de novo protein synthesis is required for initiation. However, the nature of this putative molecule is unknown. The replication initiator protein DNA a capital A is a possible candidate, and factors which promote methylation of ORIC-C and the DNA A promoter could also be involved. QV origin of replication, DAM methylation. Partition and cytokinesis. The partition of the replicated chromosome marks the culmination of the chromosome cycle and is followed by cell division. A septum forms at the midpoint of the parental cell, which is identified by a periseptal annulus, a region of modified cell envelope where the inner and outer membranes are joined together around the circumference of the cell. Additional annuli form by duplication and migrate to positions equivalent to 1/4 and 3/4 cell lengths. And these are the steps of septation in daughter cells during the next round of cell division. Once the septum has formed, the cell undergoes cytokinesis. It divides the boundary fission. The identification of mutants which disrupts cell division or partitioning has shown that the two processes can be unhitched, and such mutants fall into several categories. FT's mutants are deficient in septum formation and thus form filaments that are temperature sensitive, hence the name. The filaments often contain regularly spaced nucleoids, indicating that replication and partitioning mechanisms are still functioning normally. Minim mutants generate septa too frequently, resulting in the formation of many cells, which are small cells which contain no chromosomal DNA, although they may contain plasmids. Finally, PUR mutants form the normal-sized cells but fail to partition the chromosome properly, so that diploid and anucleate cells arise with high frequency. The pathway controlling cell division and partition has yet to be determined in full, but several key players have been identified. A good candidate for the initiation of cell division is FTSZ. This protein is structurally and functionally similar to tubulin, which forms the contractile ring in eukaryotic cells. It is distributed ubiquitously during most of the cell cycle, but is localized around the annulus at the beginning of the D phase as a Z ring. Its abundance appears to correlate exactly with the frequency of cell division. Thus, FTSZ mutants fail to form septa and generate filaments, whereas overexpression causes the production of too many septa and hence many cells. The ZIP-A protein may be important for the localization of FTAZ because its N-terminus is membrane associated and its C-terminus interacts with FTAZ. It is unclear how the septum is positioned in the cell, although nucleation sites probably exist because filaments resulting from temperature-sensitive FTAZ mutations rapidly form contractile rings at regular intervals when shifted to the permissive temperature. Genes of the mini-B locus limit septation to the central annulus and suppress the process at the terminal annuli remaining from previous cell divisions. Min-C and Min-D are septation inhibitors, whereas Min-E antagonizes Min-CD activity. The correct balance of all three products thus inhibits the terminal septation but protects the central annulus from inhibition. The partition of the bacterial chromosome proceeds similarly to plasmid partition QB. Partitioning may involve the association of chromosomes with the cell membrane and may be regulated by replication. The origin and terminus of replication as well as active replication forks are membranes associated. Bacterial mutants which affect partition fall into two categories. Those which interfere with the separation of interlocked replication chromosomes. These include topoisomerase and extra site-specific recombinase mutants, and those which affect the partitioning process itself. In the latter category, no cis-acting sites have been found in the chromosome, but several trans-acting factors have been identified. Examine the membrane protein MUKA and the microtubule-associated protein MUKB. Mutation in both genes generate anucleate cells. The next subtopic is the eukaryotic cell cycle. The standard eukaryotic cell cycle. This standard eukaryotic cell cycle is divided into 4 non-overlapping phases. The discrete events of the chromosome cycle, DNA synthesis, and mitosis occur during the S phase and the M phase, respective, and in most cell cycles, these landmarks are separated by G1 and G2 gap phases, during which mRNAs and proteins accumulate continuously. The process of crossing from one phase of the cell cycle to the next phase is a cell cycle transition. Before that, let's just have a look at this figure. This is a standard eukaryotic cell cycle as shown in the figure, right? The chromosome cycle is divided into 4 stages, G1, S, and G2, which constitutes the interphase 1, and M, which is mitosis. The left panel shows typical relative durations of the cell cycle stages, although this varies in different species and depending on cell type and growth conditions. Animal cells can withdraw to the quiescent state, GO, if growth factor are withdrawn in early G1. But once the cell passes the restriction point R, it becomes committed to a further round of DNA replication and division. The graph compares the accumulation of continuous components and with the discontinuation synthesis of DNA, the quantity of all cell components is halved at the end of the M phase when cell division occurs. Okay, and continuing on to the chapter proper. The process of crossing from one phase of the cell cycle to the next phase is a cell cycle transition, whereas mitosis is a dramatic event that involves visible reorganization of cell structure. The rest of the cell cycle is unremarkable to the eye and is termed the interphase. Variations on the theme, table 2.2, which is given here and later, include cell cycles where one or both gap phases are omitted, or whether either the S phase or the M phase is omitted, leading to halving or doubling of the DNA content, respectively. In addition, a cell may be arrested indefinitely or permanently delayed at any stage of the cell cycle, as occurs during oocyte maturation and post-mitotic cells such as neurons. Animal cells may withdraw from the cell cycle altogether, entering a quiescent state termed G0, where both growth and division are repressed. This reflects a continuing requirement for both growth factors and other signaling molecules in the environment and imposes an extra level of regulation on the cell cycle so that the growth and division of individual cells can be coordinated in the context of a multicellular organism. The abnormal cell proliferation seen in cancer is caused by the failure of this regulatory mechanism. The process of crossing from one phase of the cell cycle to the next is a cell cycle transition. Whereas mitosis is a dramatic event that involves visible reorganization of cell structure, the rest of the cell cycle is unremarkable to the eye and is termed the interphase. Excuse me. Variations on the theme, Table 2.2, include cell cycles where one or both gap phases are omitted, or where either the S phase or the M phase is omitted, leading to halving or doubling of the DNA content, respectively. In addition, a cell may be arrested, indefinitely or permanently delayed at any stage of the cell cycle, as occurs during oocyte maturation and in post-mitotic cells such as neurons. Animal cells may withdraw from the cell cycle altogether, entering a quiescent state termed G0, where both growth and division are repressed. This reflects a continuing requirement for growth factors and other signaling molecules in the environment and imposes an extra level of regulation on the cell cycle so that the growth and division of individual cells can be coordinated in the context of a multicellular organism. The abnormal cell proliferation seen in cancer is caused by the failure of this regulatory mechanism. I just want to read this table to you, variations on the theme of the four-stage eukaryotic cell cycle. Modifications. Stages omitted, no gap phases, circumstances. Rapid alternation between M and S phases is characteristic of early development in animals with large eggs because there is enough material in the egg for rapid cleavage divisions without cell growth. Many organisms miss out one or other of the gap phases. Dictyostelium discoideum replicates its DNA immediately following mitosis. Now G1. Whereas S. cerevisiae undergoes mitosis directly after DNA replication. No G2. No S phase. 2 rounds of division without intervening DNA synthesis occurring during meiosis. No M phase. Multiple rounds of DNA synthesis without cell division occur in Drosophila secretory tissues to produce polythene chromosomes. Stages extended indefinite arrest at G1, G2, or M. Withdrawal from the cell cycle G1, in brackets G0. Oocytes and egg sites may be arrested at G1, G2, or M depending on species. Fertilization releases the block and allows the cycle to resume. Many animal cells can withdraw from the cell cycle at G1 and enter a quiescent state, often termed G0, which may last months or years. This occurs if essential growth factors are withheld during G1. It involves a disassembly of the cell cycle control mechanism. Quiescent cells can be persuaded to re-enter the cycle if growth factors are made available, but there is a long delay before the initiation of the S phase while regulatory components are resynthesized. Normal somatic cells often enter G0 after a characteristic number of divisions, the Hayflick limit, a phenomenal termed senescence, which may be related to telomere length, QB, in some animals and can also be induced by certain plasmids in fungi. Some cells withdraw entirely from the cell cycle as part of their differentiation and become postmitotic, example neurons and muscle cells. And the next ensuing topic would be cell cycle checkpoints. The primary function of the cell cycle is to duplicate the genome precisely and divide it equally between 2 daughter cells. For this reason, it is important that the events of the cell cycle proceed in the correct order, and that each stage of the cell cycle is complete before the next commences. The DNA content remains constant only if DNA replication alternates with mitosis. If If mitosis occurs after the completion of DNA replication and replication commences after mitosis, is precisely divided the DNA. The cell meets these criteria by organizing the cell cycle as a dependent series of events. Thus, if mitosis is blocked, the cell cycle rests at the M phase until the block is removed. It does not go ahead and replicate the DNA anyway. That is, DNA replication is dependent upon the completion of mitosis. Similarly, if DNA replication is prevented and the cell does not attempt to undergo mitosis, because mitosis is dependent upon the completion of DNA replication. A further function of the cell cycle is to coordinate the chromosome cycle with cell growth, and so there is no progressive loss or gain of cytoplasm and no cell proliferation in an unsuitable environment. Progress through the cell cycle is thus also dependent upon cell size and is regulated by nutrient availability, the presence of mating pheromones in yeast, and the presence of growth factors and hormones in animals. The cell possesses a number of regulatory systems which can sense the progress of the cell cycle and can inhibit subsequent stages in the event of failure. These regulatory mechanisms are termed cell cycle checkpoints and represent intrinsic signaling systems of cell cycle control. The checkpoint mechanisms also respond to external signals so that arrest may occur in cases of nutrient deprivation or growth factor withdrawal. There are numerous checkpoints in the cell cycle which are clustered in two major groups: those occurring at G1 and regulating entry into the S phase, and those occurring at G2 and regulating entry into the M phase. This clustering suggests that intrinsic and extrinsic signals may funnel into common components of cell cycle regulation. Additional checkpoints ensure the orderly and dependent series of events which comprise mitosis. Different organisms attach varying degrees of significance to the G1 and G2 checkpoints, reflecting the stage at which the cell receives input from the environment. The G1 checkpoint is predominant in the budding yeast Saccharomyces cerevisiae, where it is called start, and in animal cells where it is called the restriction point or commitment point. The yeast assesses nutrient availability and the presence of mating pheromones during G1, whereas animal cells respond to the presence of growth factors. Cells of both kingdoms will arrest at this checkpoint if the environment is unsuitable for growth, but once past it, they are committed to a round of DNA replication and mitosis regardless of their environment. Conversely, in the fission yeast, Schizosaccharomyces pombe, the environment is monitored at the G2 checkpoint, and under satisfactory conditions, the cell will undergo mitosis, division, and the next round of DNA replication before checking again. The advantage of pausing at G2 rather than G1 for the haploid yeast cells reflects the presence of two copies of the genome at GP, allowing any damage to DNA to be repaired by recombination. Studying cell cycle regulation. Two complementary approaches have been used to characterize and isolate the regulatory components of the cell cycle in the heterokaryon approach. Nuclei at different stages of the chromosome cycle are joined in a common cytoplasm, and their behavior observed. Cultured mammalian cells and amphibian eggs have been used for these experiments. The results of using cultured fibroblasts synchronized at different cell cycle stages as shown here. So here this table enumerates such details, so let me just read it out for you. Fusion, S into G1, as a result of both nuclei replicate, and conclusion, S nucleus contains an S phase promoting factor. S into G2, S phase cell completes replication. G2 phase nucleus waits for S phase nucleus to complete replication, and then both cells enter the M phase. Conclusion, G2 nucleus cannot respond to S phase activator. A re-replication block, S phase activator, is also an inhibitor of mitosis. M into G1, S or G2. Interphase nucleus enters precocious mitosis regardless of state of chromosome replication. M nucleus contains an M phase promoting factor. G1 and G2, neither nucleus undergoes replication or mitosis. Both S phases and M phase activators are present transiently. The ability of M phase cells to induce mitosis in any interphase nucleus provides early evidence for the existence of an M phase promoting factor. Similar results were obtained in Xenopus nuclear transplantation studies. Interphase nucleus formed spindles when injected into eggs arrested at the metaphase of meiosis 1, and cytoplasm from these eggs could induce meiosis in oocytes arrested at G2. The large size of the eggs was exploited to purify the substance responsible, which was called maturation promoting factor. Further studies showed that maturation promoting factor could also induce mitosis in somatic cells and was in fact identical to M phase promoting factor, which shared the same acronym MPF. The second approach has been to exploit the versatility of yeast genetics to isolate conditional mutants for cell cycle functions. Numerous CDC mutants, cells division cycle, have been identified which are blocked at various stages of the cell cycle, yet continue to grow. A second class of so-called V mutants allows precocious transition of cell size checkpoints and are similar than wild-type cells. Many of the genes identified from CDC mutants are not specific cell cycle regulators but control processes such as DNA replication, repair, and mating, upon which the progress of the cell cycle depends. However, a number of CDC genes appear to play a direct role in the regulation of the cell cycle, as discussed in the following section. Satisfyingly, the biochemical analysis of MPF has shown that both approaches have converged on the same small group of molecules. The molecular basis of cell cycle regulation: cyclins and cyclin-dependent kinase, CDK. The sequential stages of the cell cycle reflect alternative states of phosphorylation for key proteins which mediate the different cell cycle events. The cell cycle transitions represent switches in those phosphorylation states. The G1 to S transition involves the phosphorylation of proteins required for DNA replication, whilst the G2 M transition involves the phosphorylation of proteins required for mitosis. The basis of cell cycle regulation is a family of protein kinases which phosphorylate these target proteins and hence coordinate the different activities required for each transition. The involvement of protein kinases in cell cycle control was revealed when analysis of S. cerevisiae, CDC, mutants blocked at start identified the product of the CDC28 gene, a T34KD. The CDC2 gene, which played an equally important role in the G2M transition in S. bombae, was found to encode a homologous protein kinase. Genes encoding similar kinases were subsequently isolated from vertebrates, and this could restore wild-type cell cycle function to yeast CDC mutants. Significantly, the Xenopus homologue of CDC28 bar CDC2 was found to be a component of MPF. The kinases were found to be present constitutively in the nucleus, but to control cell cycle transitions, the activity would have to oscillate. An explanation for their periodic activity came from the study of sea urchin eggs, wherein were discovered a family of molecules whose synthesis and activity oscillated with the cell cycle. These molecules were termed cyclins, and they were subsequently found in many other eukaryotes, including yeast and vertebrates. The second component of MPF was found to be a B-type cyclin. The activity of MPF residues in the catalytic kinase subunit, but it is dependent upon the cyclic subunit, which introduces a conformational change in its partner to stimulate kinase activity. The cell cycle kinases are thus described as cyclin-dependent kinases, CDKs, and function as CDK cyclin holoenzymes. This strategy of cell cycle regulation appears to be conserved throughout the eukaryotes. CDK cyclin diversity in the yeast and animal cell cycles. The second approach has been to exploit the versatility of yeast genetics to isolate conditioning mutants for cell cycle functions. Numerous CDC mutants cell division cycle have been identified which are blocked at various stages of the cell cycle, yet continue to grow. A second class of so-called V mutants allows precocious transition of cell size checkpoints and are smaller than wild-type cells. Many of the genes identified from CDC mutants are not specific cell cycle regulators, but control processes such as DNA replication, repair, and mating upon which the progress of the cell cycle depends. However, a number of CDC genes appear to play a direct role in the regulation of the cell cycle, as discussed in the following section. Satisfyingly, the biochemical analysis MPF has shown that both approaches were converged on the same small group of molecules. Right? The molecular basis of cell cycle regulation, cyclins and cyclin-dependent kinases, CDK. Sequential stages of the cell cycle reflect alternative states of phosphorylation for key proteins, which mediate the different cell cycle events. The cell cycle transitions represent switches in those phosphorylation states. The G1S transition involves the phosphorylation of proteins required for DNA replication, whilst the G2M transition involves the phosphorylation of proteins required for mitosis. The basis of cell cycle regulation is a family of protein kinases which phosphorylate these target proteins and hence coordinate the differing activities required for transition. While I read this, please make it a point to refer to the transcript, right? So you get to know whatever is being read out and explained. All right. If you have any doubts on those portions, do connect with me on the comment section. Okay, continuing. The involvement of protein kinases in cell cycle control was revealed when analysis of S. cerevisiae CDC mutants blocked at start identified the product of the CDC28 gene, a 34 kD protein kinase, as the principal regulator of the G1S transition. The CDC2 gene, which played an equally important role in the G2M transition, is S. Pompe. I'm reading this out because I feel I've left out some points, was found to encode a homologous protein kinase. Genes encoding similar kinases were subsequently isolated from vertebrates, and these could restore wild-type cell cycle function to yeast CDC mutants. Significantly, the Xenopus homologue of CDC28 by CDC2 was found to be a component of MPF. So that much was read, right? I'll finish up the last portion as well. The kinases were found to be present constitutively in the nucleus, but to control cell cycle transitions, the activity would have to oscillate. An explanation for the periodic activity came from the study of sea urchin eggs, which were discovered a family of molecules whose synthesis and activity oscillated with the cell cycle. These molecules were termed cyclins, and they were subsequently found in many other eukaryotes, including yeast and vertebrates. The second component of NPF was found to be a B-type cyclin. The activity of NPF resides in the catalytic kinase subunit, but it is dependent upon the cyclin subunit, which introduces a conformational change in its partner to stimulate kinase activity. The cell cycle kinase are those described as cyclin-dependent kinases, CDKs, and function as CDK cyclin holoenzymes. This strategy of cell cycle regulation appears to be conserved throughout the eukaryotes. CDK cyclin diversity in the yeast and animal cell cycles. A number of potential CDK have been isolated from yeast, but only CDC2 in S. cerevisiae and CDC2 in S. pompe appear to be directly involved in the cell cycle and are required for the GI-S and the GIM transitions in both species. In animal cells, there is a greater diversity of CDKs. The first to be discovered, the P34CDC28/CDC2 component of MPF, appears to function specifically at the G to M transition. 10 or more further CDKs are present in animal cells. 5 of these are involved specifically in the early stages of cell cycle. The diversity of cyclins is greater than that of CDKs, as different cyclins are synthesized at different stages of the cell cycle in both animals and yeast. There are at least 8 families of vertebrate cyclins designated A to H. Since CDKs phosphorylate different targets at each cell cycle transition, cyclins are required not only for kinase activity but also for substrate specificity. In animals, alternative cyclins may be differentially expressed in different cell types, which would facilitate the unique aspects of cell cycle control in distinct differentiated cells. There are generally 3 types of cyclin in all organisms: the G1 cyclins, which regulate the G to S transition; the H-phase cyclins, which are required for DNA replication; and the M-phase cyclins, which are required for mitosis. M-phase cyclins include the S-cerevisiae CLB cyclins, the vertebrate A and B type cyclins, and the S-Pombe cyclin. They are stable proteins but share a conserved motif called a destruction box, which is required for targeted ubiquitination, resulting in degradation during mitosis. Other cyclins are inherently unstable because they carry a pest domain, and their levels are determined primarily by the transcriptional activity of their genes. All cyclins carry a conserved motif. The cyclin box, which is required for CDK binding. The yeast and animal CDKs and cyclins involved in cycle regulation are summarized in the table below. This diagram shows the domain structure of cyclins. Cyclins which possess a pest motif are targeted for proteolytic degradation and are very unstable. This includes the S-cerevisiae, CLN cyclins, and vertebrate cyclins of classes C, D, E, and F. Most of these are G1 S cyclins. Mitotic cyclins tend to be stable throughout interphase, but contain a destruction box required for their ubiquitin-dependent degradation. During M phase, the first cyclins were isolated on the basis of the oscillating activity, but several are known to be synthesized constitutively and are defined on the basis of cyclin box homology rather than expression parameters. Principal CDKs and the cyclins active at each stage of the yeast and mammalian cell cycles. Please take a transcript of this. a screenshot, and then make your notes accordingly. A regulation of CDK cyclin activity. Cell cyclin transitions are characterized by bursts of CDK activity which cause sudden switches in the phosphorylation states or target proteins responsible for cell cycle events. Sudden spikes of kinase activity are not regulated by cyclin synthesis and degradation alone, as cyclins accumulate gradually in the cell, and only the mitotic cyclins are degraded by rapid targeted proteolysis. The CDK cyclin systems are active in G1 of the mammalian cell cycle. CDK to cyclin E complex is required for the G1S transition. And the other CDKs and the D cyclins are responsible for interpreting growth factor signals for the environment and act at the restriction point to channel cell into either late G1 or G0. The mammalian CDK1 cycle B complex is MPF. The vertebrate homologue of yeast, CDC2/CDC28, may be termed CDC2 or CDK1. CDK7 by H cyclin, which is a CAK. The mammalian CDK 309161 kinase is thought to be expressed constitutively. CDKs phosphorylate target proteins but are themselves also regulated by phosphorylation. The yeast CDC28 and CDC2 CDKs are phosphorylated on key residues, tyrosine 15 and threonine 161. Phosphorylated threonine 161 is required for kinase activity, whereas phosphorylated tyrosine 15 is inhibitory and dominant to threonine 161 phosphorylation. The principal determinant of CDK cyclin activity. Regulation of CDK cyclin activity. Cell cycle transitions are characterized by bursts of CDK activity which cause sudden switches in the phosphorylation states of target proteins responsible for cell cycle events. Sudden spikes of kinase activity are not regulated by cyclin synthesis and degradation alone, as cyclins accumulate gradually in the cell, and only the mitotic cyclins are degraded by rapid targeted proteolysis. Two CDK cyclin systems are active in G1 of the mammalian cell cycle. The CDK2 cyclin E complex is required for the G1S, and the G1 is a transition. The other CDKs and the CD4 cyclins are responsible for interpreting growth factor signals for the environment and act as the restriction point to channel the cell into either late G1 or G0. The mammalian CDK1 cyclin B complex is MPF. The vertebrate homologue of yeast CDC1 by CDC28 may be termed CDC2 or CDK1. CDK7 by H cyclin, which is CAK, the mammalian CDK 30161 kinase, is thought to be expressed constitutively. CDK is phosphorylated target proteins, but are themselves also regulated by phosphorylation. The yeast CDC28 and CDC2 CDKs are phosphorylated by two key residues, tyrosine 15 and 30161. Phosphorylated 30161 is required for kinase activity, whereas phosphorylated tyrosine 15 is inhibitory and dominant to 30161 phosphorylation. The principal determinant of CDK cyclin activity in yeast is thus the state of phosphorylation of tyrosine 15, and some of the upstream regulatory components have been identified. In S. Pombe, V1 is a tyrosine kinase which phosphorylates CDC2 at tyrosine 15 and thus inactivates it. V1 activity is antagonized by CDC25 phosphatase, which removes phosphate groups from the same substrate. Both V1 and CDC25 are themselves regulated by intrinsic and extrinsic signals. This is believed to be the basis of the G2M checkpoint in the species. The decision to proceed with mitosis or RSG in G2 thus reflects relative levels of these opposing activities, as regulatory networks which feed into the checkpoint are considered below. An enzyme has been identified in mammals which is responsible for 30161 phosphorylation. And remarkably, this turns out to be yet another CDK cyclin complex comprising CDK7 and cyclin H. It is known as CDK activating kinase, CAK. The enzyme responsible for 301,4 phosphorylation has not yet been identified. CDK cyclin complexes are also regulated by inhibitory proteins. This third level of control is used for both intrinsic and extrinsic regulation purposes, as shown here. The S-Pombe RUM1 protein is a specific inhibitor of the mitotic CDK cyclin complex and is synthesized throughout the G1 and S1 phases, thus preventing cycle skipping DNA replication and entering mitosis prematurely. The FAR1 protein is activated in response to mating type pheromones in S. cerevisiae. Regulation of CDK cyclin activity. The enzyme responsible for 301,4. This third level of control is used for both intrinsic and extrinsic regulation purposes, as discussed below. The S-Pombe RUM1 protein is a specific inhibitor of the mitotic CDK cyclin complex and is synthesized throughout the G1 and S phases, thus preventing the cycle skipping DNA replication and entering mitosis prematurely. Excuse the incoming telephone, it's been cut off. And the FAR1 protein is activated in response to signaling by mating type pheromones. And S-cerevisiae and inhibits the start CDK cycling complex, thus arresting the cell at G1 in readiness for mating. Two families of CDK cycling inhibitors, CDK1s or I's, are found in animals. One blocks all CDK cycling activities and the other specifically inhibits these cycling complexes containing CDK4 and CDK6, whose major substrate is a retinoblastoma protein. These regulatory mechanisms are discussed in more detail here. Progress through the cell cycle. Transition of the start checkpoint in yeast. Start is the predominant checkpoint in the S-cerevisiae cell cycle and is well characterized in the species, figure 2.4. In GI, the CDC28 CDK can associate with any of three cyclins, CLN1, CLN2, CLN3, which are functionally redundant, at least under laboratory conditions. The genes can be deleted singly or in pairs with no effect on phenotype. With mutation in all three arrests the cycle at G1. The highly unstable CLN3 is expressed constitutively and acts as an indicator of cell growth. When the concentration of CDC28 CLN3 reaches a critical level, the kinase activates two transcription factors, SBF and MBF, which contain a common component, SW16. SBF initiates transcriptions of the genes of the other G1 cyclins, CLN1 and CLN2. These cyclins associate with the CDC28 to form CDK cycling complex with several functions. And the complexes with several functions related to the activation of these 6 CLB cyclins, the mitotic cyclin CLB1, CLBZ, CLB3, and CLB4, and the SV cyclin CLB5 and CLBG, which share a conserved destruction box. Firstly, they reduce the rates of proteolysis of the CLB cyclins, and secondly, they increase the rate of hydrolysis of a CDK CLB cyclin inhibitor, SIC1. The second transcription factor, MBF, initiates transcription of the CLBS and CLB6 genes, thus by stimulating the transcription of SV cyclins and increasing their stability. The G1-specific CDK cycling complex induces the onset of the S-phase when cell growth is sufficient. The S-phases, specifically CDK cycling complexes, stimulate the transcription of activity of replication proteins, perhaps including those found at the constitutive origin, recognition complexes on yeast ARS elements. This is a summary of the molecular regulation of start in S-cerevisiae. Accumulation of CLN3 acts as an indicator of cell size. Dashed line. At a critical concentration, the kinase activity of CDC28 is induced and activates two transcription factors. SPF promotes CLN1-3 transcription and generating a positive feedback loop, although proteolytic degradation of CLN2 due to its starvation can delay entry into the S-phase. MBF activates the genes for CLN5 and CLN6, whilst activated CDC28 kinase inhibits destruction, box-targeted cyclin degradation, and promotes degradation of the inhibitor. SIC1, CLN5, and CLN6 form complexes with CDC28, which activates the expression of replication genes. The cell cannot pass start until mitosis is complete, and in S. pombe, this is achieved by a simple negative feedback mechanism in which the mitotic CDK cycling complex inhibits a protein required for the initiation of DNA replication, CDC18. In the absence of active CDC18, DNA replication is blocked, and this repression is lifted when the mitotic kinase is destroyed. Entry into the S-phase in mammalian cells. In mammals, four CDKs, CDK2, CDK3, CDK4, and CDK6 act in early GI, all four associate with these D cyclins, which are synthesized early in GI in a growth factor dependent manner. CDK4 and CDK6 play the principal roles in the regulation of downstream events. The only known target of these early CDK cycling complexes is the retinoblastoma protein RB1, which is a negative regulator of the cell cycle. RB1 in its unphosphorylated form binds transcription factors of the ED2 family, which normally activate genes required for entry into S-phase. RB1 inhibits the expression of these genes in two ways: by sequestering the E2F activation domain. The only known target of these early CDK cycling complexes is the retinoblastoma protein RB1, which is a negative regulator of the cell cycle. RB1 in its unphosphorylated form binds transcription factors of the E2 family, which normally activates genes required for entry into S-phase. RB1 inhibits the expression of these genes in two ways: by sequestering the E2F activation domain, the direct repression by chromatin remodeling. Phosphorylation RB1 by the early G1 CDK2 cycling complexes releases the repression. An important downstream effect of CDK D-cyclin activation is the synthesis of E-cyclins, which are required for the GIS transition itself. The E-cyclins form complexes with CDK, and these activate CD525A phosphatase, which may in turn activate the S-phase specific complex CDK2-A cyclin. This complex is required for the initiation of replication and has been localized at replication regions where it can be immunoprecipitated with pCNA, a component of the major eukaryotic DNA polymerase. Its specific targets in the initiation complex remain to be characterized, but both cyclin A and cyclin E complexes are known to phosphorylate and regulate the activity of several transcription factors, including the E2F family, P53, BMYB, and the inhibition helix loop helix protein ID2. The restriction of replication at once per cell cycle. The cell fusion described above show that the S-phase nucleus can induce the GI nucleus to replicate, but not the G2 or M nuclei, which need to complete mitosis before becoming competent for replication once again. In all cases, replication occurs only once. A model proposed to explain these data involves a replication license factor displaying the following properties. It interacts with DNA origins in the latter stages of mitosis. It is essential for initiation of replication, but is inactivated or destroyed once initiation has taken place. It is unable to cross the nuclear envelope. The model proposes that a cytoplasmic licensing factor interacts with the DNA origins before reassembly of the nuclear envelope at the telophase. This factor is a target for regulation by S-phase promoting CDK cycling complexes and initiates replication at each origin at the beginning of the S-phase, becoming inactive in the process. During the remainder of the cell cycle, the new licensing factor is unable to enter the nucleus and the precautious initiation of replication is prevented. The factor enters the nucleus at the start of mitosis but is inactive or prevented from interacting with DNA because of the condensed state of the chromatin. The factor interacts with the origin as the chromatin decondenses, but remains inactive, awaiting the S-phase signal. The nuclear envelope closes, preventing entry by further licensing factor and any uncomplex factor in the nucleus, and perhaps also in the cytoplasm, is targeted for degradation. Proteins which display many of the properties of the licensing factor have been identified in xenopus, mammals, and yeast. In S. cerevisiae, the product of the CDC4/6 gene is thought to be the licensing factor or a component of it, although in yeast the nucleic envelope does not break down during mitosis, and the licensing factor is transported into the nucleus by translocation. Control of entry into mitosis. In yeast, entry into mitosis is controlled by the same cyclia-dependent kinase that regulates start CDC2, CDC28, but in combination with specific mitotic cyclins. In S. cerevisiae, these are encoded by the CLB1, CLB2, CLB3, and CLB4 genes, and in S. bombay by the CDC13 gene. In vertebrates, The CDK-CDC2 kinase associates with A and B type cyclins, which, like their yeast counterparts, contain mitotic destruction boxes. Much of the regulatory pathway of the G2/M checkpoint has been determined in S-Pombe. Similar components are found in other eukaryotes. Essentially, mitosis is initiated by a burst of CDC2 activity generated by a post-translational positive feedback mechanism. This differs from the entry into S phase in S. cerevisiae, which is governed by a transcriptional feedback loop. The mechanism is broadly conserved throughout the eukaryotes, but the checkpoints for entry into mitosis are regulated in the different ways as discussed below, which will be dealt with in the next lecture. Thank you.